This is BSR. Today we are going to discuss about biomolecules, purely NEET oriented. So question wise we are going to discuss. So the question, look at there. Watson and Crick proposed certain features for double helical model of structure of DNA. Which of the following is not a feature of double helical structure of DNA? The question is very clear. Now look at the options. Option 1, two chains are anti-parallel to each other. And the second one, nitrogen bases of two strands are complementary and paired through covalent bonds. And the third option, each DNA strand is made up of nucleotides. Fourth option, nitrogen bases are present towards inner side of double helix. So there are four options. Question is very clear. Which of the following is not a feature of double helical structure of the DNA? You can say easily. Very important. Look at the second option. Nitrogen bases of two strands are complementary and paired through hydrogen bonds. Adenine complementary with the thymine, cytosine complementary with the guanine. Adenine complementary with the thymine by 2 hydrogen bonds, cytosine complementary with the guanine by 3 hydrogen bonds. Next question, which of the following is mismatched? So, option 1, chitin polymerase, it means polymer of glucose amine. Second option, glycogen is a polymer of glucose. And the third option, cellulose heteropolysaccharide. Fourth option, inulin homopolysaccharide. The question is very clear. It's a mismatched. Chitin clearly polymer of glucosamine. Glycogen is clearly polymer of glucose. Cellulose is a homopolysaccharide. Here the third option, it is not heteropolysaccharide, so it is a mismatched, clearly third option. Then you go for the next question. Inulin is a polymer of glucose, fructose, sucrose, maltose. So the four options are there in the third question. You can say very easily fructose. Clearly inulin is a polymer of fructose. Then next question, look at that. Which of the following elements are present in a negligible amount in human body and very little amount in earth crust? There, C, O, N, S, Na, Ca, Mg, Si. Carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium and silicon. First option Cn, SiOCa, third option Sin, fourth option magnesium, silicon. First option carbon, nitrogen, second option silicon, oxygen, calcium, and the third option silicon, nitrogen, fourth option magnesium, silicon. Yes, the third option silicon and nitrogen. Then next question. Read the following statements. A. Fatty acids are saturated or unsaturated. Fatty acids are saturated or unsaturated. Melting point B, look at there. Melting point of oils are less. Hence they remain a liquid in winter. C. The molecular weight of liquid does not exceed 800 daltons, but they are present in acid insoluble pool. Then D. Glycerol is a compound lipid which is trihydroxypropane. So which statements are correct? So A, B, C, D, 4 statements are there. In that, which statements are correct? ABD, ABC, third option ABCD, fourth option AB only. Yes, first one. Fatty acids are saturated or unsaturated? Yes. Melting point of oils are less, hence they remain liquid in winter. Yes. Second statement also correct. The molecular weight of the lipid does not exceed 800 daltons, but they are present in acid insoluble pool. Correct only. They are present in acid insoluble. You know that one. The D, glycerol is a simple lipid. Glycerol is a simple lipid. So, they are given compound lipid. So, except D, A, B, C is correct. A, B, C is correct. Okay. Then, we are going to next question. Read the following statements. Each protein is a polymer of amino acids. A protein is a heteropolymer and not a homopolymer. Dietary proteins are the sources of essential amino acids. Hemoglobin is the most abundant protein in whole of biosphere. How many the above statements are right? How many statements are right? Definitely hemoglobin is the most abundant protein in human being, but not whole biosphere. Rubisco. So except D. In my point of view, A, B, C are right correct statements except D. So in my point of view, the second option is correct. In everybody point of view, second option is correct. Okay. Then go for next question. In sea urchin, DNA which is double stranded, 17% of the base were shown to be cytosine. The percentage of other three base to be present in DNA are. When the cytosine percentage is 17 in the sea urchin, DNA is double helical. You know, definitely when the cytosine percentage is 17, guanine percentage is also 17. 17 plus 17 is equal to 34. 34%, remaining 66%, definitely, A 33%, thymine 33%. So, my point of view, second option is correct. Guanine 17%, adenine 33%, thymine 33%. Then, go. The next question. A DNA has 10 nucleosomes. Find out the number of phosphodiester bonds and hydrogen bonds in the DNA if adenine percentage is 15. So, good question. 10 nucleosomes are there. Then, how many phosphodiester bonds and how many hydrogen bonds are there in the DNA when adenine percentage is 15? Is a very important clue to you. You calculate the phosphodiester bonds, always n minus 1. N is nucleotide minus 1 on one strand. When two strands are there, minus 2. So here definitely two strands. When the two strands, double-stranded DNA is there, you go for n minus 2. When only one strand is there, you go for n minus 1. Yes, 10 nucleosomes. In each nucleosome, how many base pairs are present? 200 base pairs. 200 base pairs means definitely in each nucleosome 400 nucleotides are present. 400 nucleotides, 10 nucleosomes. So 10 into 400 nucleotides means 4000 nucleotides. When 4000 nucleotides are there in one strand, 2000, in another strand 2000, total 4000. So 4000 minus 2, it means how many phosphodiester bonds are present? 3998. Clear? Yes. Next point. When you go to 4000 nucleotides are there. 4000 nucleotides or 4000 nucleotides means how many base pairs are there? 2000 base pairs. 2000 base pairs or 4000 nucleotides. In one coil, 20 are present. In one coil, in one DNA helix, 20 nucleotides are present. when 4000 nucleotides are there, when 4000 nucleotides are there, how many coils are there? 4000 by 20. 4000 by 20. It means how many coils are there? 4000 means definitely 200. 200 coils are there, it is very clear. But here important thing, adenine percentage is 15. When adenine percentage is 15, definitely thymine percentage is also 15. When adenine and thymine 15 is there, definitely cytosine and guanine percentage is 35 and 35. Suppose when you take only one coil, adenine percentage is 15 means definitely 20 into 15 by 100. It means 3 adenines are complementary with 3 thymines. 35% means 20 into 35 by 100. It means 7. 7 cytosines are complementary with 7 guanines by 3 hydrogen bonds. So 7 cytosines are complementary with 7 guanines by 3 hydrogen bonds means 7 into 3 equal to 21 hydrogen bonds. Their 3 adenines are complementary with 3 thymines by 2 hydrogen bonds means 3 into 2 equal to 6. 6 plus 21 is equal to 27. In one coil, 27 hydrogen bonds are there. In one coil, 27 hydrogen bonds are there. 27 into 200 coils are there. 27 into 200 coils are there. It means how many hydrogen bonds are present? 5400 hydrogen bonds. So my option is second one. Okay. Then when you go for next question, which one is incorrect match? First option, alkaloid, morphine. Second option, toxin, resin. Third option, lectin, curcumin. Fourth option, pigments, anthocyanin. So which one is the incorrect? Alkaloid, morphines and codians. You know that one. Toxins, resins and abrin, you know. Whereas the pigments, anthocyanin and carotenoids also there. Whereas lectin is concanavalin. Remember. So curcumin is not a lectin. Okay. Then answer is definitely incorrect one is lectin, curcumin. Okay. Then how many peptide bonds present in a 900 amino acids contain polypeptide chain? How many peptide bonds present in a 900 amino acids containing polypeptide chain? Polypeptide chain is a protein. Suppose when 3 amino acids are there, 1, 2, 3. Here one peptide bond, here one peptide bond. So how many peptide bonds? 2 peptide bonds, 3 amino acids. When 3 amino acids are there, 2 peptide bonds. So definitely n minus 1. When 900 amino acids are there, definitely 899 peptide bonds are present. Next, macromolecule in a cell include all except. Macromolecule in a cell, macromolecule in a cell include all except nucleic acid, DNA, first option. Nucleotide, second option. Third option, RNA. Fourth option, protein. You know, nucleic acids, proteins, all are macromolecules. Nucleotide, nitrogen bases are micromolecules. You know that one. Yes. Then, next question. Statement 1. Each protein is a polymer of amino acid. Definitely. All amino acids assemblage to form a protein. Yes. Then statement 2, amino acids are substituted ethanes. Statement 2, amino acids are substituted ethanes. No, it's not ethane, it's a methane. So, my point of view, statement 1 is true and statement 2 is false. So, the third option is correct for this question. Yes, then. The next question. Biomolecules which are found in the acid insoluble fractions are called biomacromolecules. Yes, definitely. Whatever biomolecules are there. Statement 1, look at there. Biomolecules which are found in the acid insoluble fractions are called biomacromolecules. Correct. Statement 2, polysaccharides are long chains of sugars. Yes. Statement 1 is true and statement 2 also true. Two statements are true. So definitely first option is correct. A and true. A statement is 1 is true and statement 2 is also true. So the next one. Glucose is stored in human body in which of the following form? Glucose, direct question. That is the glycogen. First option. No doubt at all. Glycogen, collagen, starch, sucrose. Answer is first. Then next question, look at there. A list of different metabolites is given below. Find out the number of primary and secondary metabolites respectively. You know the difference between primary metabolites and secondary metabolites. Primary metabolite having a role directly in physiological process. Secondary metabolites are having economic importance. So look at their alkaloid, heparin, ricin, carotenoids, and lectins and drugs. These are secondary metabolites. Alkaloids, heparin, ricin, carotenoids, lectins and drugs economically useful to human being. So they are secondary metabolites. Whereas amino acids, glucose, fructose, fatty acids, thiamine and uracil, these are primary metabolites. So 6 and 6. In my point of view, answer is second option. 5, 6, 6, 6, 7, 5, 4 and 8. So answer is 6 and 6. Next. Which one is the correct average composition of a cell respect of percentage of total cellular mass? Which one of the following correct average composition of a cell in respect of percentage of total in cellular mass? Water, protein, carbohydrate, lipid, nucleic acid and ions. Definitely water composition 70 to 90%. Protein 10 to 15%, carbohydrate 3%, lipid 2%, nucleic acid 5 to 7% and ions 1%. Definitely my point of view, first option. Yes. Then you go for next question. Among the following, which one is the sweetest sugar? Direct. Fructose is the sweetest sugar. No doubt. Sucrose, glucose, fructose, maltose. In that point of view, fructose is the sweetest sugar. Then next question. A nitrogenous base is linked to the pentose sugar. Nitrogen base linked to the pentose sugar by. Already we discussed that one. N-glycosidic linkage. That bond is called N-glycosidic linkage or bond. Okay. So definitely the second option is correct. First option phosphodiester linkage, N-glycosidic linkage and ester linkage. Phosphoester linkage. Answer is N-glycosidic linkage. Next, which of the following group belongs to acid insoluble? Insoluble. Okay. First option, proteins insoluble, nucleic acid insoluble, polysaccharides insoluble, lipids also insoluble. No doubt, my point of view, first option. No doubt. Whereas when you go to second option, lipid, nucleic acid, fatty acids and amino acids. Nucleic acids are micromolecules. Third option, nucleotides, proteins, polysaccharide, lipids. Nucleotides, micromolecules. Proteins, monosaccharides, nitrogen bases, lipids. Monosaccharides and nitrogen bases are micromolecules. So first option. So, the given below is the structural formula of sucrose, ribose, glucose, deoxyribose. Yes. Difference between ribose sugar and deoxyribose sugar. Definitely ribose sugar is having two functional groups, whereas in deoxyribose sugar only one functional group. Directly you can go for ribose sugar. Yes, then you go for next question. In double stranded DNA molecule, each step of ascent is represented by a pair of bases. At each step of ascent, the strand turns. Directly the angle between one DNA helix to another helix angle directly 36 degrees. Directly 36 degrees. No doubt. Answer is second option. Yes. Next question. The length of one coil of bDNA helix is bDNA. Length of one coil. One coil length 34 angstrom units or 3.4 nanometer by 10. 34 angstrom units or 3.4 nanometer length of the coil. 34 angstrom units or 3.4 nanometer. Whereas the distance between one nucleotide pair to another nucleotide pair is 3.4 angstrom units or 0.34 nanometer. I think I am clear. Okay, then. Which of the following statement is incorrect for DNA? Look at the options. Yes, DNA is a long polymer of deoxyribonucleoside monophosphate. Correct. Bacteriophage known as 5x174 has 5386 base pairs, 5386 nucleotides. Haploid content of human DNA is 3.3 into 10 power 9 base pairs. Correct. 5-methyl-uracil nitrogenous base present in DNA. First option is correct. DNA is a long polymer of deoxyribonucleoside monophosphate. Yes. Second option, a bacteriophage known as 5x174 has 5386 nucleotides. So second option is wrong. Okay. Next, which of the following is the most abundant protein in the whole of biosphere? Already came that question. Actually, the answer is rubisco. It is present, it is abundant in plants. So the next question. The monomer unit of fungal cellulose is first option, mannitol, N-acetyl glucosamine, and the third option, galacturonic acid, ascorbic acid, direct, N-acetyl glucosamine. Yes, next question. At the time of protein synthesis, which group of first amino acid is involved in peptide bond formation? Yes, definitely. Carboxyl group of first amino acid, you just observe there. Always peptide bond formation takes place between carboxyl group of first amino acid and amino group of second amino acid. It is very clear. So definitely at the time of protein synthesis, which group of first amino acid is involved in the peptide bond formation? Definitely the first option, only COH. No doubt, don't go for other options. Second option, only NH2, both COH and NH2, both alcoholic. So definitely first option, no doubt at all. Then, next, look at that. The enzyme trypsin hydrolyzes. First option, proteins to peptides, fats to fatty acids, glucose to ethyl alcohol, polysaccharides to monosaccharides. Hydrolyzes, catalyzing the hydrolysis of ester, ether, peptide, glycosidic, CC bonds, C halide or PN bonds also. It's hydrolyzing the ester bonds, hydrolyzing the ether bonds, peptide bonds hydrolyzing, glycosidic bonds are also hydrolyzing, CC bonds also hydrolyzing by the hydrolysis. So definitely proteins to peptides answer, first option. Okay, then next, Which of the following secondary metabolites is a toxin? Codeine, monoterpenes, concanavalin A, ricin. Oh, 1, 2, 3, 4. Memory based. Codeine is alkaloid, monoterpenes, terpenoids, concanavalin A is a lectin. Definitely, it is a toxin is ricin. Toxin, ricin. Important, simple question, but you should remember. Next question. The lowest molecular weight compound in acid insoluble fractions are? Lowest molecular weight. That is insoluble fraction. Present in insoluble. First option, proteins, no. Nucleic acids, no. Polysaccharides, no. Fourth option, lipid. Next question. The cell wall of fungi are composed of chitin cellulose, no. Cellulose and polysaccharides, no. Chitin and polysaccharides, correct. Chitin, polysaccharides and glycogen, no glycogen. Answer is chitin and polysaccharides, no glycogen. No glycogen. Okay. A molecular biologist analyzed a nucleic acid sample and found that it contained the nitrogen bases in the following proportion. Adenine percentage is 20, thymine percentage is 15, cytosine percentage is 35, guanine percentage is 30. This data indicates that a molecular biologist analyzing their adenine percentage is 20, thymine percentage is 15, cytosine 35, guanine 30. According to Chargaff, you know, definitely adenine complementary with the thymine by 2 hydrogen bonds. Here adenine is 20%, thymine is 15%. It means there are no two strands. Definitely it is a single stranded. Definitely it is a single stranded. Whether it is a DNA or RNA, definitely thymine is there. Definitely it is a DNA. So nucleic acid in the sample is single stranded DNA. It is not a double stranded. It is a single stranded. My point of view, first option is correct. Don't go for any other option. Should not go. Biomolecules, these are all useful, purely NCERT based questions. Level 1 questions and up level 2, level 3. It means linked questions also we discussed, and almost all we discussed. Further, I will give more and more best questions to you. Thank you to all.